It's my great pleasure to introduce Olaf Sporns. So if anyone's been doing anything in the neuroimaging space, they've probably heard the term connectome, and he and his colleagues coined that term, I think, back in 2005. And
20 years, yeah.
Yeah, so he's the one to blame for the term. And I like your sort of comment about sort of short. It's like got a long tail, I guess, of history, because one of the things is when it was coined is like a lot of things before that time were kind of rebranded as, connectome type work. So it's a sort of natural way to describe some of the things we're doing, but it can also be abused. So there's a lot of pros, a lot of issues to rich space to kind of talk about. So Olaf is a distinguished professor and provost professor in psychological and brain sciences and the Robert H. Schaffer Chair at the University of Indiana. And I will, without further ado, pass the baton to you, Olaf. And thanks again. I'm looking forward to hearing your talk.
Thanks so much, Vincent and Armin, for inviting me, and great to be with you today. I'll get right on with it because we have only so much time. And good morning, good afternoon, good evening to the audience as well. So as Vince mentioned, one of my interests in the last 2 or 3 decades has been brain connectivity and connectomics, of course. And I want to, in my talk, cover a few of the key concepts that I keep coming back to in my own work, and then also highlight a few of my more recent efforts in this area. So let's get right to it. Obviously, the last 10, 20 years have been extraordinarily productive in terms of research carried out in non-human species, in invertebrates, and in rodent brain and non-human primates, in both structure and function in the area of brain connectivity. And it's been remarkable to see this area grow and mature and really explode over the course of my career. When I first got started, brain connectivity was very arcane, was not really something that was mainstream or thought of as being mainstream. And to see this grow so explosively has been really quite an experience for me personally. So in my talk, I'll cover a little bit about connectomics and brain architecture, mostly in structural connectivity or anatomy in the beginning. Then I'll pivot over to functional connectivity and a little bit about functional connectivity dynamics and communication. And hopefully at the end, I'll have a chance to talk briefly about some recent work that I'm very excited about on information theory applied to the brain. So let's get right to it. Connectomics really is defined on both structural and functional connectivity networks, and the structure-function dichotomy really is fundamental in most biological systems. When I first got started in the field, we knew about the human brain mostly from post-mortem histological dissection, not an ideal experimental vehicle, as you can imagine. But then about 20 years ago, we got the first images coming from diffusion imaging and tractography, which opened a new window on brain architecture, on brain connectivity, on anatomical connectivity in the human brain non-invasively. Pretty soon, working with Patrick Hagman at the time, we started building the first sort of connectome networks from these data, connections between brain regions in the human brain, in the human cerebral cortex for the most part. And simultaneously, there was the rise of resting state, as it became called very, very quickly, spontaneous fMRI fluctuations and cross correlations between them, which were then called functional connectivity. So all these concepts kind of emerged in sequence and gave rise to a very rich field of covering both structural and functional networks. Both of these constructs can be very productively described using tools from network science, broadly conceived. On the left, a structural network, a sparse a graph of connections in the human brain, in this case, symmetric and weighted connections. On the right, a similarity network or correlation matrix, covariance matrix, functional connectivity, also coming from the human brain. As you can see, these two types of data are very interrelatable with each other, but I would like also to point out that they are very different mathematical objects, and we have to be a little careful when we analyze and model these 2 different types of data, that we use the appropriate tools for that purpose. When we got started with Patrick Hagman, one of the initial discoveries was that there were some features of connectivity in the human connectome that stood out to us. Every region had its own unique set of inputs and outputs. There was a broad, heavy-tailed degree distribution, high clustering and short path length, the two hallmarks of small worldness. And we found evidence for modules interlinked by hub regions, and the hubs themselves weren't isolated, but also shared connections with each other, more so than expected by chance, a feature that we initially called a structural core, later on became called a rich club. Those features, which stood out to us even in our very first grand total sample of 5 individuals in our initial paper, really, I think, have been found again and again in many replications since then, and seem to be ingrained as large-scale features in the human connectome architecture. Not only that, but also animal connectomes that have been constructed since then with very different techniques, from track tracing to fluorescence microscopy to EM reconstruction. Those connectomes from non-human animal species have also shown similar features of unique fingerprints, modules, and hubs. So there are common, shared, universal sort of features of structural connectomes that are shared across different species, which stood out to us as an interesting finding, right? I mean, an interesting fact that connectomes are not necessarily reflecting very specific features of each species, but have some common architecture to them. So that posed the next question for us. I'm working with Larry Swanson and Joel Hahn. I've been working for the last 10 years, really, systematically going through the rat nervous system, starting at the time in 2015 with the cerebral cortex, and then working our way forward into many subdivisions that you can see illustrated at the bottom. And finally now in the brain and the central nervous system, and then by next year in the nervous system. So that work built on the collection of track tracing data published in the rat anatomy literature that Larry and Joel are meticulously collecting and annotating. From that, they build directed connection matrices of hundreds of regions, as you can see for the CNS, almost 800 regions and about 72,000 connections between them. And we then proceed forward with analyzing these connectome data sets with the tools of graph theory and network science, and have done so systematically, really, for all these different subdivisions that you see on this slide. One of the things that stands out to me is that while we in our field in fMRI especially, we are focused on cortex a lot in humans, the cortex is a very small part of the brain in the rat, about 150 regions out of 790 total. And it turns out that when you look at other subdivisions of the brain, the thalamus or the tectum or the spinal cord, the types of connectivity that you find are very characteristic, are not at all identical to the ones that you see in cerebral cortex. So I think when we talk about the brain and the connectome, we need to pay attention to the fact that there's many different subdivisions of the brain, not just the cortex, and that each one of them will have its own sort of connectivity fingerprint that will tell us a little bit about what kind of information processing problems it might be involved in solving. So there's a lot of heterogeneity across these different subdivisions, and our over the years have tried to express that. Secondly, one of the main constructs in our work has been modularity, which I still think is one of the most fundamental properties of connectome organization. Now, modularity is often computed on human brain networks and other networks, but there's a twist, which is that for many real-world networks, there isn't a single partition that fits best. There are many partitions that are almost equally good at explaining the modular architecture of the system. What we use for our purposes in this line of work, and we are since using it in all of our work, including fMRI, is a hierarchical approach to capturing modularity, not at one level of scale only, but at really all accessible levels. And then computing from that what we call a co-classification matrix, which is for each node pair, the likelihood or the frequency with which that node pair is found in the same module across different size scales. This gives us a graded assessment, which incidentally is quite related to functional connectivity. And it gives us a graded assessment of the modularity architecture of the system. We can then decompose that into a modular hierarchy, which in our work in RAT, the modules actually quite often correspond to known functional circuits that are subserving specific behaviors in the RAP. So there's a methodological advance that came out of this line of work, really, that we have now imported in other work as well, a characterization of modularity in a graded fashion across multiple scales, which I find much, much more informative than settling on a single partition as if it's the only one that matters to a system. That's a little bit of an aside. Where does this universality of architectural features in the connectome of multiple species, where does that come from? And here I think back to some work I did many years ago now with Ed Bullmore, where we thought about the issue of cost and the issue of efficiency and the trade-off between these two. The idea is sort of like this. If the brain was shaped largely by constraints of cost, let's say energy consumption or conduction delay or wiring length, things like that, then all connections in the brain should be optimally short, right? Because that minimizes volume and cost the most. So then we end up with a brain that looks like a lattice. Of course, that brain is extremely inefficient in moving information around. So there's an opposing trend which wants to create potential paths of communication between many, if not all, pairs of regions in the brain. That, however, leads us to extremely expensive architecture where connections are far-flung and long distance all over the place. That's not sustainable from the issue, from the constraint of cost. So there's a trade-off we hypothesized where the brain is largely cost conserving, that is, connections are kept short, that results in spatially compact modules. And then there's hub regions, not many of them, but a few, which maintain long distance connections that link the modules together and make sure that integration can happen. So there's a trade-off between cost and efficiency. That was really more like a theoretical framework back in the day. And going forward, we recently had a chance to take a look at comparative connectomics in a more extensive sample of species under homogeneous conditions of imaging. And that was with the work that we've done recently with Yaniv Asaf and Yossi Yovel at Tel Aviv University. They had amassed a very impressive connection of hundreds of specimens from of various mammalian species, over 100 species in total, that had come to them from collections in the wild or from collections from zoological gardens where animals had passed away of natural causes. As you can see here, we had in our hands through them connectome samples from animals as widely diverse as the leopard, the wild boar, the fruit bat, or the capybara. If you don't know what a capybara is, it's a very large rodent, one of the largest rodent species out there. So we set about analyzing these connectomes for differences, commonalities, common features across these 100 different mammalian species. The main construct that we used was the co-classification matrix, which is our graded assessment of modularity, as I mentioned earlier, across many, many scales. That tells us something about the propensity of each node pair of being in the same module. So that's a richer assessment of modularity across these connectomes. The first step was taken by my former grad student, Josh Faskowitz. Josh took genetic information from these 100 species and built a phylogenetic tree. And that allowed us to not only build a tree of how these species are related to each other, but also a similarity assessment of their genetic similarity or phylogenetic similarity. He then went on and operationalized a measure of connectome distance, which is a distance measure where we can quantify without having necessarily node correspondence, where we can quantify how similar two networks are to each other. Then asked a very simple question, and one that we hoped the answer was going in the right direction. And that is, if we look at two animal species that are within the same taxonomic order, let's say they're both rodents, they're both carnivores, they're both primates within the mammalian system, will they have more similar connectomes to each other than pairs of animals between different taxonomic orders, like a rodent and a bat compared to each other? And indeed, this is a validation test. Within taxonomic order, species pairs showed more similar connectome architecture. So that was more like validation. That was more like something we hoped to find to underscore that the imaging and the reconstruction, which is quite laborious, actually gave us something that we could take forward into further analysis. The next step then was to look at how brain size played into connectome architecture. Why brain size? We immediately noticed that even across the mammalian a branch of phylogeny, where all animals are somewhat related to each other, we see huge differences. In fact, 3 orders of magnitude differences in brain volume, from the smallest bats to the largest carnivores and primates. 1,000-fold difference in brain volume. And we know from lots of allometric studies and comparative evolutionary analysis that brain size plays an important role in also shaping other aspects of brain architecture, like gray matter, white matter volume, and connectivity. So what Josh then did is he took off the shelf an existing generative model by which we could essentially, through a generative process of probabilistically assigning connections, we could try to rebuild these connectomes, where we have one free parameter, which is the fall-off with distance or connection density. When that parameter is large in magnitude and negative, it means that as connection distance increases across the brain, connections are becoming increasingly sparse. And the severity of that effect is expressed in the magnitude of that parameter. If the parameter is negative 4.5, it means that distance factors into connectome topology a lot. If it is smaller, like negative 2, then distance does not play such a large role. Fitting this model to connectomes of different animals from different species and with different brain sizes, there's a very clear relationship of how this parameter scales. And for the larger brains, the distance penalty is very severe. For the smaller brains, the Chiroptera, for instance, the bats, all the way on the left, the distance penalty is much less severe. That suggests that indeed geometric size, the size of the brain, actually plays an important role in addition to phylogenetic distance in shaping connectome architecture. My former postdoc, Maria Grazia Pucheru, then took the next step, and that was to look at modularity and the co-classification matrix across these different species in more detail. And just one nugget from her very extensive work published last year in PLOS Biology is the following: that as brain volume increases, once again, the larger brains are on the right side of the x-axis over here, the relationship between an existing connection weight, and the co-classification magnitude, that is the propensity for those 2 nodes that are connected to also be in the same module, increases a lot. What you're looking at here is a relationship that strengthens as brains get bigger, meaning that if we find a connection of a certain weight, it is highly predictive of whether the connected nodes, the connected brain regions, are also in the same module or not, and vice versa, of course. So once again, geometry and connectivity are interconnected and are intersecting in this metric of modularity and co-classification magnitude. I'm skipping many results from Maria's paper here, but at the end, what we're looking at is a schematic where, summarizing the main findings of the paper, we're seeing as brains get bigger in size, modules become more well-defined, more offset from each other in terms of topology, and they also become denser, internally denser, more strongly connected with each other internally, and spatially more compact, which places the connections that link up these modules over long distances at very high premium in the brains of animals that have in animals that have large brains. So even outside of looking at phylogenetic and evolutionary trends, the increase in brain volume that we see across the mammalian taxonomic branch is very informative about how topology plays out. So it's not all about adaptation in the end. It's also about geometry and geometric factors is sort of what we take away from this line of work. So going over to dynamics and functional connectivity, as you all know, the brain can be measured in terms of resting state. Functional connectivity can be constructed from resting state fMRI recordings. And a question that we've been pursuing for many years in the lab is how do we get from something that is relatively spatially sparsely connected brain that is relatively static over time, at least on the time scales of a resting state scan. How do we go from that to something which is as dynamic, as ever-changing and even turbulent as the picture that you see on the right, functional connectivity as it unfolds over time? What's the relationship between these two domains? As you all know, we're building resting state networks from, taking individual frames of a resting state scan, computing a cross-correlation matrix, and then clustering it basically, where the modules that we get are recognizable systems that now have names such as default mode, dorsal attention, limbic, motor motor, visual, and so forth, reliably found across individuals, across different studies. It's worth remembering that this was a real sea change in the way cognitive neuroscience was kind of formulated and conducted. Those systems did not exist 20 years ago, at least not to the same extent. Default mode was around to some extent, but not as well known. But now really a lot of our discussion about, how the brain is organized and how it changes in disease or across the lifespan is couched in terms of these very reproducible resting state systems that we are now so familiar with. And they really come from ultimately really an assessment of modularity of resting state functional connectivity, static functional connectivity, that is Pearson correlation over all measurements. I emphasize static because it seemed clear to many of us, I think Vince was very much in the lead on this early on as well, that while we can compute static functional connectivity, when we look at smaller and smaller segments of time, there was also a lot of variability of functional connections. And, I wrote a little think piece here some years ago about how we might conceptualize dynamic functional connectivity as an ebb and flow of more segregated states that are more modular, where the modules are more well-defined and offset from each other, and other states that are more functionally integrated. And so this interplay and ebb and flow of segregation integration seemed one way to describe how functional connectivity might be dynamic and how we might conceptualize these dynamics in our analyses and our models. This idea was very fruitful, and lots of work has been done since then by Andrew Zaleski, as well as Max Schein, Russ Poldrack. There's so many things. I mean, of course, Vince's work was even preceded this, I believe, with Allen et al. In 2013. The idea that we can, through windowed analysis, we can find episodes of functional connectivity that actually differed from each other. There was some non-stationarity present in the data. We also pursued windowed analyses for a while with my former postdoc, Makoto Fukushima. And a small snippet from one of his papers here is that he actually tracked modularity, the well-known Q metric, over time, essentially computed for each window at a given moment, and then through an appropriate null model, filtered out moments in time or windows where the Q metric was particularly high, unexpectedly high, and unexpectedly low. When you average those moments together, you get recognizable functional connectivity signatures for those high Q or high modularity moments, which across two different data sets essentially look like a disconnection between what I might call loosely extrinsic systems and intrinsic systems. That is, systems that are more sensory motor on the one side and attentional, and on the other side, default mode and frontoparietal. So this signature was something that we found across different data sets. And while the minimum Q states were much more heterogeneous, the windows were much more different topologies that could not be easily classified and actually gave rise, on average, to a sort of flat, what you might call a more integrated state. For some years, we worked with windowed analyses like this, and quite frankly, I was not very happy about them because you kind of in the worst possible, you're kind of in a bad intersection of having moments that are not really moments, but still 30-second, 60-second segments of a resting state recording, so not momentary. And you have functional connectivity estimates that are actually quite uncertain because you have very few measurements that you can work with. And there's 3 parameters involved that no one is quite comfortable with, like window size, overlap, things like that. So we were a little bit uncomfortable, which gave rise to a period of creative turmoil in the lab. We looked for ways to get to dynamics in a slightly more straightforward way, and perhaps zooming in on the more momentary fluctuations, the fluctuations that occur at single TR resolution in fMRI. And Rick Betzel, my colleague at the time, and Josh Fastowitz, my grad student, came up with the following idea, which was, I think, quite nice and has given us a lot since then. When we're computing Pearson correlation, we take 2 node time series, we normalize them, z-score them essentially, and then look at their cost correlation with each other, which actually is nothing else but the moment by moment, TR by TR product of the 2 time series, and then the average of that. And we thought, Rick and Josh really had the idea first, to leave it at the edge time series and essentially use the co-fluctuation time series of the node pair as our basic construct. So omit the averaging step. If you take 2 node time series, you multiply them together like this, you end up with an edge time series. And if you omit the averaging step, you can now take this co-fluctuation time series forward into other analyses. If you do the averaging, though, then the average of this construct is equal to the mean of the mean of this time series is equal to the functional connectivity. So that was a very interesting idea because there are no free parameters involved. It was just taking functional connectivity and going one step back instead of averaging, retaining all of the edge time series, and then performing analyses on that. Edge by time, this is an edge by time matrix that you see here. If we take that matrix and we average it along the y dimension, we get a vector. If that vector then gets folded back into square form, we are getting functional connectivity, which is the construct everybody has been using and is still using today, static FC. We noticed that there were these vertical streaks in the edge time matrix, and initially it gave us something to worry about because anybody in the fMRI space is always worried about potential artifacts, physiological artifacts, head motion, and things like that. It turned out that these moments of high co-fluctuation that manifested in these vertical streaks in the matrix did not have any obvious relationship to physiological artifact or head movement. We labeled them at the time events because they stood out as singular point-like discontinuities in the time course of the fMRI run. These events, as it turns out, are universally found in virtually all recordings we've looked at from different imaging centers, different conditions, including rest, task, and movie data. And they struck us as being somewhat conspicuous, certainly. It turns out that they are part of a continuous distribution. They are sort of on a long tail of, as you can see in this diagram here, the long tail of a distribution of amplitudes. And they happen to be moments that are more representative of the static functional connectivity, I think for mathematical reasons, because they have larger amplitude, they factor into the calculation more. There's lots of things that we can do with edge time series and events. I want to just show you a movie I made of a typical resting state run coming from the Human Connectome project. I show this in all of my talks for a reason. On the upper left-hand corner is the functional connectivity matrix, which is the construct everybody in our field is familiar with the familiar systems that you can see indicated: visual, motor, dorsal attention, ventral attention, limbic, frontal horizon, default mode. The movie that's playing in the middle is the momentary co-fluctuation pattern that unfolds for 1 TR at a time. And the mean of all of those frames in the movie is exactly equal to the functional connectivity shown on the left. So it is, in some ways, a very natural decomposition of the static functional connectivity into single TR contributions that we can access and display, as you can see here, and track over time, for example, in terms of their amplitude characteristics, as you can see in the plot at the bottom. Sometimes the amplitude is very high. That means there's high co-fluctuation overall in the system. Sometimes it's reasonably low. I show this because at no moment in time do you really see the complete, functional architecture on the left displayed, right? You see co-fluctuations sometimes obeying the confines of one of these little cubes or rectangles along the main diagonal. But most of the time you see systems forming, dissolving, linking with each other in certain combinatorics, and disappearing. And that ebb and flow of co-fluctuation is something that I still think is a very, very rich construct to mine for individual differences, for temporal and dynamic characteristics. And we have since, working also with Rick, of course, and others in the field, taken this forward in many, many different directions. And Rick, if nothing else, has taken this in many more directions than we have. Just a couple more points about this. A little snippet of interesting insight, I think. On the top, you see, once again, the edge time series and that matrix I showed you earlier. Once again, if you take the mean along the y dimension, you fold it up into square form, you get functional connectivity. No surprise here. At the bottom, what I've done is I've thresholded at 0. So now the time series have been binarized. Edge pairs are, node pairs are either co-fluctuating together or not. If they co-fluctuate together, the system encodes it as a 1. If it's below, it's a 0. So if we take that construct where we've totally thrown away all of the amplitude information from the run, we can actually compute an agreement matrix, because it turns out this is actually a community vector that amounts to a bipartition of the whole network into 2 co-fluctuating ensembles at each TR. If we build the agreement matrix from that construct is virtually identical to functional connectivity. So we can reconstruct something which resembles, with very, very high accuracy, 0.97 correlation, the full functional connectivity from a binarized representation of our run, where we've actually eliminated all of the amplitude information. That to me is still not surprising necessarily. I think there's a mathematical reason for it, but it is, I think, another fruitful way of thinking about our recordings, because we can take this binary construct and we can do things with it that are actually otherwise inaccessible, because we now have single TR decomposition of a continuous process into discrete samples, and that is a very very interesting way of doing analysis and modeling. One of the upshots from this is that, yes, functional systems are not permanent and permanently present, if you wish, but are a statistical construct, and they are expressed intermittently when co-fluctuations are high or low. And so they blink in and out of existence, as it were. This allows us to do lots of interesting statistics on which systems are evident when, and how these systems combine with each other to co-express or not. So there's a rich temporal signature of systems, functional systems coming in and out of existence, if you wish. That information can be used for purposes such as fingerprinting, Emily Finn's work, or expressing brain behavior relationships, not based on full functional connectivity anymore, but on subsets of moments that when combined give us very strong signals. In fact, as Sarah Katz, my former PhD student, now postdoc at Yale, has shown that using sort of machine learning techniques, you can extract moments in time that will increase signal to noise, if you wish, in fingerprinting and brain behavior relations significantly. And that is another very important, very interesting line of work that we've been pursuing in the lab. Another couple of points I want to make. You might ask about these events. What happened to them? I haven't mentioned them in a while. Well, when we look at movie data, I should say this is the work of Vic Betzel's lab and Jake Tanner especially. It turns out that not in all cases, but in some, these events actually will synchronize across subjects. This suggests that they're not, stochastic measurement artifacts or even perhaps obvious physiological artifacts, but perhaps are in some ways driven by or triggered by stimulus information. Secondly, very recent work carried out with Mike Miller and Mike Zaniga at UCSB. Through them and collaborators in Germany, there was access to patients who had undergone calosotomy because of untreatable epilepsy. Looking at resting state functional connectivity post-calosotomy, there were several interesting findings in that sample, one of which I want to highlight here. This is a patient at the top who has had a partial calosotomy with some of the splenial fibers at the back of the callosum spared, while the patient at the bottom had a complete or full calosotomy. When we have spared fibers connecting the two hemispheres, the edge time series that we construct separately for the left and right hemisphere display dynamics that are highly correlated. The correlation is on the order of 0.7, 0.8. When the callosum is completely cut, the left and right hemispheres become desynchronized. The edge time series become desynchronized. It's not just lowering functional connectivity across the two hemispheres. That is certainly also true in that case. But it's also the temporal signature of these fluctuations is now very much less synchronous than in the case when the callosum is intact or when fibers are spared. This suggests to me very strongly that in fact the fluctuations that we see across time are indeed orchestrated by underlying structural connections, in this case the callosum. To drive this point home in a modeling context, Maria Pope, former PhD student, now postdoc at University of Chicago, took off the shelf a model that we had worked with years ago, the Kuromoto model, a coupled oscillator model that allows us to simulate large-scale dynamics and also use with a simulated HRF function, essentially produce simulated fMRI responses. We were interested in figuring out whether that model would also display rich temporal dynamics at the time scale that we observed in the empirical data. It turns out it does, without any parameter addition or any additional, without messing with the equations at all. The Kuromoto model gives us edge time series that have the same sort of characteristic streakiness to them, which means high co-fluctuation events occur. They occur intermittently, somewhat infrequently, and they have very similar phenomenology to the ones that we see in the empirical data. So despite the fact that there's no active process driving these fluctuations, causing them to appear, they do appear just as a result of the dynamics that occur within the system. Now, a very important factor, as Maria's work showed, for creating events was we needed a modular structural connectivity matrix. So now here we are looping back to the first part of the talk where I talked about modularity as a fundamental architectural feature in many animal connectomes. When we offered the model a modular SC matrix, such as the one coming from the actual human brain, we saw events and fluctuations of some magnitude very readily. when we offered the model a non-modular structure connectivity, same number of connections, but no modules, there were no events. These large excursions did not occur. We traced these events to ultimately to what people in physics call cluster synchronization, the synchronization of events that involve elements that are bound together in connected clusters within the connection matrix. It's actually a phenomenon that's reasonably well studied and understood in physics, but we did not know about it. So cluster synchrony, the existence of structural connectivity patterns that have modularity are critical for these events to occur. Secondly, we can even trigger them, injecting highly synchronized phase synchrony actually into the Kuramoto model in clusters. And then we're getting high cofluctuation magnitude events right afterwards with the simulated fMRI response. All of this, the model, the empirical work I showed you with the colostotomy, to me drives home the point that when we see these fluctuations in our fMRI recordings, these single TR fluctuations that I showed you a lot of in this talk, those are not stochastic, measurement artifact or what have you. They actually are, it's the architecture talking to us. It's a modular architecture that synchronizes in certain patterns driven by structural connectivity that is actually giving rise to these partly organized, partly disorganized states that we can pick up with this decomposition of classic FC into single TR contributions. A brief note on a whole other line of work that we've been pursuing in the lab for many years now, that is network communication. When you have a sparse network like the brain and two regions want to pass information to each other, it creates a difficult problem of how they might connect, because they're not usually directly connected through a direct pathway. Instead, information has to travel over a multi-step path. Multi-step paths are common, very numerous actually, in sparse networks. But the shortest path, which is the most efficient way of passing information with the fewest number of steps along the way, is actually very hard to find. In technological systems, in network analysis, we need to use pretty powerful algorithms to actually access the shortest path. The brain doesn't really know where the shortest path is. It doesn't have those algorithms. It doesn't have a map of its own topology that it can scrutinize and plot. So there's a problem here. How do brain regions communicate with each other efficiently, quickly, when the demand arises in a sparse network where not every pair of nodes is directly connected? The problem can be reformulated in the following way. Starting from a structural connectivity matrix, the connectome on the left, we may implement a particular policy by which information is passed between nodes, for example, accessing the shortest path if we so choose. And then that gives us for each pair of nodes a matrix that we might call a communication matrix that tells us how easy or how difficult it is to pass information from one node to the other. And that communication matrix can then be compared to, for example, functional connectivity as an outcome of neuronal couplings. That approach has been taken by several of my postdocs actually over the years, most recently by Kaio Seguin, who did the following very ingenious modeling study accessing, I'm going to first talk about Joaquin, my postdoc some years ago now professor at Purdue, who started this line of work really in my lab. And just a small snippet from his work about 10 years ago, looking at a metric called search information, which quantifies the amount of information that's needed in a given network for a source to access a shortest path to a target. It turns out that search information, which is coming again from the physics literature, really primarily navigation of complex networks by agents, search information is actually quite informative, very highly correlated with empirical functional connectivity, much to our surprise really at the time. On the left, functional connectivity empirically measured in the resting state fMRI. On the right, predicted functional connectivity from actually a compound measure of communication metrics. The nice thing about this approach is you can calculate the communication matrix analytically. You don't have to actually do a whole full-up simulation of the brain. You can actually do matrix algebra and some information theory on it, and you can get a prediction, essentially a prediction of functional connectivity that's quite accurate, given how simple the approach is. So that line of work was taken forward by Caio. Finally, I can talk about his work here, utilizing a data set that was publicly shared from a large amount of work done on patients that had been implanted with depth electrodes in the cortex pre-surgically in preparation for epilepsy surgery. What the surgeons did is pass the electrical currents from a stimulation electrode to the other electrodes to record how information was flowing in those patients' brains, in part to figure out pre-surgically something about their brain organization. These data consisted of thousands, if not hundreds of thousands of experiments, really, that recorded latency and amplitude for given stimulation and recording pairs across the human brain. What Caio did is utilize these data, put underneath a structural connectome that we now possess for the human brain, imperfect as it may be. We have a version of it available to us at this time. And that can then be coupled with what I mentioned earlier, communication protocol or a policy, such as shortest path efficiency or search information or communicability, each of which operates somewhat differently and makes different predictions about which sources and which targets are most likely to connect. We can compare the resulting communication matrices to the existing empirical measurements of how sources and targets communicated with each other in this invasive stimulation experiment setting. Using that approach and comparing these two domains with each other, Caio was able to essentially rank how well do different communication models fare in this competition. Turns out search information, which I mentioned earlier from the work of Joaquin Goni, is one of the best ways of predicting communication patterns or functional connectivity in the human brain. Communicability, purely based on diffusion of information, not shortest path at all, comes in a close second, while shortest path efficiency, which is most commonly used in our field still to assess communication properties in a given brain network, comes in third. So these are ways of assessing different communication strategies that have different policies underneath, different algorithms that guide them, and comparing them to empirical data. And that is an ongoing project that will, I think, take a lot more in the future to really figure out which policy is really implemented in the nervous system. And perhaps the answer is, in the end, it's not just one policy, but a combination thereof. I quickly go to the end, just a few more minutes here, and then I'm done, to talk about information theory as a third complementary aspect of how brain systems are organized. Working with, again, my former postdoc, Maria Grazia Pucheru, we recognized at one point that we could actually come up with a new way of clustering functional connectivity data sets based on information theory, not based on edge weights or densities of connections or things like that, but based on the time series and the amount of information that is embedded in them. The idea is as follows. Functional systems are internally redundant. They're composed of regions that are somewhat alike with each other, that have highly correlated temporal dynamics. It's redundancy, same information represented across multiple nodes. These nodes then jointly constitute a functional system. We can quantify that shared information between sets of nodes as what's called high total correlation. Total correlation is a multivariate extension, as you can see in this equation at the bottom, sort of a multivariate extension of mutual information. Information is just between two pairs of regions. while total correlation is really estimated on an ensemble or on a set of regions. We can quantify this, we can compute it, and we can use total correlation in a cost function to compute a score for a given partition of the brain. How well does that partition, how well does it fare in terms of putting systems together that internally share high total correlation? When we do that, the principle illustrated here on the left, we find a partition or partitions that are not unrelated to the canonical systems we're all familiar with from the published resting state literature, such as the famous 7 Yao systems that you see at the top in this diagram. At the bottom is the optimized partition that Maria retrieved from using this fairly new, I would say, cost function based on multivariate total correlation. In the paper, there's a lot more discussion about how these patterns relate to each other, similarities and differences between them, and how we might view them across, for example, different individuals, but also across the lifespan. It's a new avenue towards information theory as applied to functional connectivity analysis that I find very intriguing, in part because 30 years ago, I worked with Tononi and Edelman on measures of complexity that were using very similar constructs. And in fact, we had gotten to this new approach on modularity through the work of Maria Pope and Thomas Varley, 2 of my former PhD students who really drove this line of work forward. And I want to go beyond redundancy for a moment. Redundancy is something that we have emphasized in our work in fMRI, especially in resting state analyses, quite a lot. Redundancy involves, well, the name says it, the representation of identical information across a set of elements. However, there's an antipode to this, or an opposite on another end of the spectrum, which is synergy. Synergy involves information that is only available, is integrated across a set of units that themselves have unique informational signatures, so not identical. I sometimes like to compare this to the redundancy to, let's say, a string section in an orchestra where all the strings play the same tune. No surprise there. The individual instruments are doing the same thing. Synergy is more like a small jazz ensemble or a string quartet where every player has their own line that they are pursuing, and the composition emerges uniquely from the interactions between the different elements, the different instruments coming together. You cannot take one instrument and decode the composition from it. It's not possible. You have to listen to the whole ensemble all at once. That's synergy. Where is synergy in the brain was the question that we pursued, Maria and Thomas pursued, and linking back to some work that, again, I mentioned I had done with Tononi and Edelman like 30 years ago. Some of this stuff is getting a little bit revived now, and that's nice to see. What they looked at was where are the subsets of a certain size in the brain that express high synergy. There's a way that we can quantify synergy, which is not shown on this slide, but we can compute it. If we look at where these ensembles are, they are not lining up with known functional systems at all. Instead, the corresponding regions reside at the interstices of these functional systems, as shown here on the right. That is, if contributions from multiple functional systems temporarily become integrated with each other, we get synergy. And along the lines of the analogy of the string quartet or the small jazz ensemble, where instruments come together and separate again, and that creates the harmony that we are so used to listening to. There's a lot more work in this area that's bubbling up these days and pursued in multiple laboratories around the world. Really high order interactions are quite up and coming these days. It's very nice to see that, quite frankly, is that we move away from just bivariate interactions, but into higher order space. And there are many questions that have been posed and can still be posed that are actually quite intriguing and subject to further work. Finally, what I've done today is I've shown you a few examples from network analysis that you've seen in the earlier part of the talk, moving this into a discussion of dynamics, a very rich area, not just in fMRI analysis, but also in general systems neuroscience, the idea of dimension reduction, synchrony, communication, and manifolds. The interaction between these two, very fruitful for modeling and analysis purposes, and then emerging on top of that interaction, the domain of information, which, if nothing else, gives us a very rich set of statistical and mathematical tools to mine multivariate higher order interactions in the brain. I also would hold out hope that this triangle, the interaction between networks, dynamics, and information, is not only fruitful for doing analyses, but it actually matters to the brain itself that these relationships exist, and it informs and guides our internal dynamics of brain networks and circuits and systems, and guides behavior and cognition. So that is my hope for the future. I've gone through a lot of material at the end. Just a brief shout out to a new journal that is not so new anymore. It started about 10 years ago. I'm stepping off by the end of the year from the editorship, and Alex Fonito is taking over in January, and I hope very much that many of you will consider it for your future work. With that, I close. Thank you very much.
Thank you. Thank you. That was wonderful.